So good morning, everyone. Indeed, it's a warm morning for all of us. February last week, last Saturday here, and it's nice to see all of you in the gathering with the august guest in the audience. Welcome to the Sir Edward Mellanby Memorial Oration and the Annual Day Function. Today we are joined by distinguished guests Dr. Ashutosh Sharma and Dr. Jyotsna Dhawan, who are not only eminent researchers but also have played important role in capacity building, community learning, mentoring students and young faculties. And there is another common thread between these two distinguished guests. They have keen eye and interest for different forms of art, design, and music. So we welcome both of you to CDRI. We are also joined by directors of sister CSIR labs. Welcome to CDRI, sirs. And we have a director from CBMR. We have teams from ISIR Kolkata, BITS Pilani Goa campus, and we have representatives from SE Biotech Private Limited. We are happy to have our former colleagues, former HODs and chief scientists who are present in the audience. And we are also joined by family members of Dr. Nityanand and Dr. VP Kamboj. So on behalf of CDRI family, I welcome you all. We will begin the program with Sir Edward Mellanby Oration, and as you all know, Sir Mellanby was a British physician and pharmacologist. His seminal research helped to establish the link between vitamin D deficiency and rickets. And in 1951, he helped to establish CDRI and served as the first director of CDRI. To begin the program, I request Dr. Radha Rangarajan and Dr. Ashutosh Sharma, Dr. Jyotsna Dhawan, and Dr. Sanjay Batra to come up on the dais. So welcome again to all the dignitaries. I would like to request Director to present the bouquet of flowers to our guests. So Dr. Ashutosh Sharma and Dr. Jyotsna Dhawan. I would like to invite Director to address the audience to begin the program.
Good morning to all of you, distinguished guests, Dr. Ashutosh Sharma, Dr. Jyotsna Dhawan, directors of CSIR sister laboratories, Dr. Bhaskar Narayan, Dr. Prabodh Trivedi, Dr. Ajit Sachne, Professor Alok Dhawan, Director CBMR, Dr. Sonia Nityanand, Vice Chancellor KGMU, family members of Dr. Nityanand and Dr. Kamboj, Director ISIR Kolkata, Dr. Soumitra Banerjee, collaboration partners from BITS Goa and ISIR Kolkata, colleagues past and present, friends from the press and media, and my dear students. I welcome you all to the 49th Sir Edward Mellanby Memorial Oration, followed by our annual day celebrations. It is a distinct honor and pleasure to have with us Dr. Jyotsna Dhawan and Dr. Ashutosh Sharma. Dr. Dhawan is Emeritus Scientist at CCMB, Hyderabad. Thank you very much for accepting our invitation to be here and deliver the Sir Edward Mellanby Memorial Oration. I welcome Dr. Ashutosh Sharma, President, Indian National Science Academy, former Secretary, Department of Science and Technology, and currently Institute Chair Professor at IIT Kanpur. Welcome. Thank you very much for being with us today. We begin the program by paying homage to two of our most iconic directors who sadly passed away within months of each other. Dr. V.P. Kamboj passed away on 21st November 2023, and Dr. Nityanand on January 27, 2024. Their loss leaves a huge void in our hearts and minds. Dr. Nityanand, a visionary scientist and institution builder, is globally known for his work on Cencroman, a non-steroidal women's contraceptive agent that was developed from concept to drug at CDRI. This remains a remarkable achievement, not only for then, but even for now. He joined CDRI soon after its founding, went on to head the medicinal chemistry division, and was director from 1974 to 1984. In his tenure, besides Cencroman, multiple drugs such as Senbrucridin and Gugulipid were also developed. But beyond CDRI, he played a huge role. He was instrumental in the setting up of the Indian Pharmacopoeia Commission, in fact, even the NIPER system, and in the framing of patent laws that recognized process but not product patents. And this really enabled the Indian pharmaceutical industry to take off, and today we are suppliers to the world. He led an extraordinary life and will be remembered for his contributions to this institute and beyond. Dr. Ved Prakash Kamboj was another visionary director of CSIR-CDRI. He joined the Division of Endocrinology in 1961 as a research scientist. He later became head of department, and he took over as director in 1992 and continued till 1997. At the time of his death, he was the chairperson of the ICMR Committee for the Product Development Center and guiding the phase 3 clinical trial of Picroliv for non-alcoholic fatty liver disease. This is a product of CDRI. So he remained active, he remained plugged in to research and, in fact, into the CDRI ecosystem all the way to the end. He was an expert in the field of reproductive biology, fertility regulation, and endocrinology. And he played a key role, along with Dr. Nityanand, in conducting the clinical trials for Cencroman. He served as the president of the National Science Academy of India from 2005 to 2006. To the families of Dr. Nityanand and Dr. Kamboj, thank you for joining us. Although your loss is unimaginable, we hope in some way that our collective expression of gratitude and acknowledgement of their contributions gives you some strength. We will now present a tribute to Dr. Nityanand and Dr. Kamboj.
I request the dignitary.
The CSIR-Central Drug Research Institute faces a profound loss with the past. The CSIR-Central Drug Research Institute faces a profound loss with the passing of two distinguished mentors, Dr. Nityanand and Dr. V.P. Kamboj. Dr. Kamboj's departure on November 21, 2023, at the age of 86 in his hometown of Jalandhar, Punjab, left all of us deeply shocked. Following closely on January 27, 2024, Dr. Nityanand left us at the age of 99 in Lucknow after battling a brief illness. Their passing is not only a loss for the Central Drug Research Institute and the Council of Scientific and Industrial Research, but is also felt deeply across the nation and across the scientific world. We mourn the loss of these two luminaries whose legacies will continue to inspire future generations. Dr. Nityanand was born on 1st January 1925 at Layalpur, West Punjab, now in Pakistan. He had his early education at Layalpur, obtained his BSc from Government College Lahore in 1943, and then moved to Delhi for MSc in Chemistry from St. Stephen's College, which he completed in 1945. He then went on to University Department of Chemical Technology, Bombay, where he obtained his PhD in 1948 under the supervision of Professor K. Venkataraman. followed by yet another PhD at St. John's College, Cambridge University, under the supervision of Professor Alexander Todd. Dr. Nityanand returned to India in year 1950, and after a short stay at Delhi University, he joined the Medicinal Chemistry Division of the newly inaugurated Central Drug Research Institute in March 1951 and continued here until his retirement in 1984 and beyond. His stay in CDRI, first as a scientist, then as head of Medicinal Chemistry Division from 1963 to 1974, and later as director from 1974 to 1984, has been of great significance in shaping and directing the institute. He has published more than 400 research papers and over 30 book chapters. He has been granted about 130 national and foreign patents and has jointly authored multiple important books. He has inspired generations of chemists and biologists in the country, and he supervised about 90 students for their PhD degree and mentored many postdoctoral fellows. Many of his students today occupy important positions in academia and industry throughout the world. Dr. Nityanand's contribution to the growth and development of the Central Drug Research Institute has been phenomenal. He propelled its advancement to a world-class center for drug discovery and development, especially for tropical diseases, central nervous system disorders, contraception, and drugs from medicinal plants, areas of special national relevance. His contribution to the nation in form of his work on Centromad is well known to all of us. He was associated with different drug policy formulating bodies of the nation for almost 4 decades and has been an advisor and consultant to many scientific bodies and institutions. He has been a member of the Scientific Advisory Committee to the Cabinet of Government of India from year 1981 to 83, member and chairman of Steering Committee for Chemotherapy of Malaria of World Health Organization and multiple other agencies. In recognition of his contributions to the nation, Government of India in year 2012 conferred Dr. Nityanand with the civilian honor of Padma Shri. He continued to be involved in scientific progress of CDRI during his service at the institute and beyond. Dr. Nityanand passed away on January 27, 2024 in Lucknow at the age of 99, leaving an indelible mark on the scientific community. His enduring legacy is characterized by his relentless pursuit of knowledge, innovation, and mentorship, inspiring countless scientists and researchers to follow in his footsteps. Dr. Ved Prakash Kamboj, a luminary in the field of reproductive biology and endocrinology, left an indelible mark on the scientific community through his pioneering research and leadership. Born on April 1, 1937 in Lamba Pind village, Jalandhar, Punjab, Dr. Kamboj's journey in science began with his foundational education in Jalandhar city, leading to a distinguished academic record at Punjab University, Chandigarh, where he earned his BSc Honours in 1959, MSc Honours in 1960, PhD in 1965, and DSc in 1970. His career at the Central Drug Research Institute, Lucknow, began in 1961 when he joined the institute as scientist, eventually becoming the head of the Department of Endocrinology in 1981. Dr. Kamboj's leadership roles expanded as he became deputy director and later led the institute as its director from 1992 to 1997. His tenure as president of the National Academy of Sciences, India, from 2005 to 2006 underscored his prominence in the scientific community. Dr. Kamboj's contributions were not confined to administrative leadership. His research significantly advanced the understanding of reproductive biology and fertility regulation. He also contributed significantly to the development of the non-steroidal contraceptive pill omeloxifene, which showcased his commitment to impactful scientific innovation. With over 300 research papers and numerous books to his name, Dr. Kamboj was a prolific contributor to the body of knowledge in his field. His accolades, including the Rambaxi Research Award in Medical Sciences in year 1992 and the Vigyan Gaurav Award by the Council of Science and Technology Uttar Pradesh in year 2000, reflect the recognition of his contributions by the scientific and academic communities. Elected a Fellow of the Indian National Science Academy INSA and holding other prestigious memberships and fellowships, Dr. Kamboj's career was marked by a relentless pursuit of knowledge and service to science. Dr. Kamboj's contributions to the Central Drug Research Institute have been instrumental in establishing it as a beacon of innovation and excellence in the field of reproductive biology, endocrinology, and drug discovery. His efforts have not only advanced the Institute's capabilities in drug discovery and development, but have also highlighted the importance of addressing reproductive health issues through scientific inquiry and development. Dr. Kamboj passed away on November 21, 2023 in Jalandhar, Punjab at the age of 86 years, leaving behind a legacy that will continue to inspire and guide future generations in the realms of science and research. His life and work exemplify a rare blend of scientific brilliance and a commitment to making a tangible difference in the world. Today, we remember Dr. Nityanand and Dr. V.P. Kamboj not only for their groundbreaking contributions to science and humanity, but also for their unwavering commitment to mentoring the next generation of scientists. Their legacies at the Central Drug Research Institute and CSIR and beyond are a testament to their extraordinary lives dedicated to the pursuit of knowledge and the betterment of society. As we reflect on their remarkable achievements and the void their departures leave behind, we are reminded of the impactful life that one individual can have in this world. Their memories will forever inspire us to strive for excellence and to continue their work in advancing scientific discovery and innovation for the welfare of humanity. The CDRI family collectively mourns the loss of these cherished mentors, and we extend our heartfelt prayers for the eternal peace of their noble souls.
So after this tribute, we will move on to the oration. The 49th Sir Edward Mellanby Memorial Oration would be given by Dr. Jyotsna Dhawan. She's an Emeritus Scientist in CSIR-CCMB. She's well known in the scientific community, but just for the students, would just give a brief biography of Dr. Dhawan. She obtained her PhD in cell biology from Boston University, and then she pursued her postdoctoral work on adult stem cells and gene therapy at Stanford University. She relocated back to India and joined CCMB in 1996. And there, her research group has made seminal contributions in understanding muscle stem cell and repair. From 2009 to 2014, Dr. Dhawan helped to establish Institute for Stem Cell Science and Regenerative Medicine, which is also called INSTEM, and it is located in Bangalore. And she served as the dean of that institute. Her lab looked at the mechanism of use of dormant stem cells to promote tissue regeneration. Dr. Dhawan's work has been recognized both nationally and internationally, and she's recipient of Wellcome Trust Senior Research Fellowship, and she's a fellow of Indian National Science Academy. She has served as the president of Indian Society for Cell Biology. And she also serves as an advisory board of Ignite Life Science Foundation, an independent effort by scientists, entrepreneurs, management professionals to support funding and to support the life science ecosystem in India. And with these few words, I would like to invite Dr. Jyotsna Dhawan, and we look forward to your talk, ma'am.
on? Am I audible? Am I audible? Yeah, okay. So it's a great honor for me to be here. And I want to thank CDRI and Dr. Radha Rangarajan for inviting me, and Dr. Batra for the wonderful arrangements. It's really wonderful to be here in this iconic institute, and I was really impressed by the beautiful tribute you put together for your two amazingly well-known directors who contributed so much, not only to this institute, but to Indian science. And so it's really a pleasure to be here. When one thinks about a person like Edward Mellanby, in whose name today's oration is there a conflict? Yeah, okay. So I just wanted to say a couple of words about the inspirational life of Sir Edward Mellanby. He's very well known to all of you, but I had no idea that in addition to the fact that he worked as this nutrition scientist, he also made such major contributions to scientific institutions, including CDRI, but also from the MRC, that he had this huge societal impact because of his efforts at nutritional policy. And that's really something, because for a man who enjoyed working at the bench and who, in fact, apparently took weekends away from his work as the Director General of the MRC to go and work in the lab. And I show here a picture of his wife and scientific partner, May Mellanby, with whom he did much of his work. I think that's a great example for us to all follow. So I guess it's not working. Maybe I can step back. Okay. So I promised you that I'd talk about dancing with quiet stem cells. Looks like I'm doing the dance. This one? This is the pointer, right? And this is the, okay, great. All right. Now that I'm done with the technological challenges, what I'm going to talk to you about in the next 35 to 40 minutes, and if I go too long, please somebody wave. I know an oration is supposed to be a much more formal occasion, but I'm an informal person and I won't be offended if somebody tells me that I'm droning on too long. So what I'm going to talk to you about is the work that we've done over the last 25, 27 years on dormant stem cells, specifically stem cells in muscle, and why the issue of dormancy has any importance at all. Okay, so muscle matters because it's can be up to 30% of body mass, except for some of us who are couch potatoes who have much more fat than muscle. But the cell biology of muscle regeneration is what we've been looking at. Muscle matters not just in healthy life, but also because it's lost in aging and disease. There are a large number of myopathies and dystrophies, which are very distressing conditions, many of them inherited disorders for which there is really no therapy, which are intractable. So the issue of why dormancy or quiescence, as its technical term is, in stem cells, a natural question might be, why do you care about dormant stem cells? And I'll tell you a little bit about what we've learned. And when I say we, one has to be sure that this is a large group of people working across the world. I will give credit to many of the pioneers in the field, and this is by no means a single-handed contribution. So as you know, muscle powers movement. Movement as sport is one of the great joys. Muscle also powers art, movement as art. Interestingly, the facial muscles, of which we have 43, are responsible for conveying emotion. So muscle is a very varied tissue. And it's also, as you would know here, a very key player in systemic metabolic regulation. So for all of these reasons, muscle has been a focus for many biologists. Just in terms of its structure and development, muscle is made up of bundles of myofibers. And these muscle fibers are multinucleated cells, which are held together by connective tissue. And internally, you have a highly structured actomyosin contractility apparatus, which actually powers movement. So damage to skeletal muscle leads to injury, and this is adult skeletal muscle in the adult, which has this beautiful regular pattern in cross-section, in a histological section. Damage leads to complete degeneration of the fibers, but 2 weeks later, the tissue regenerates completely. And the reason why it's able to do so is because of the existence of these dormant stem cells which persist from embryonic development in the adult tissue. And these were identified in the 60s by Alexander Mauro, and they're found to reside underneath the basement membrane which surrounds the skeletal muscle fiber. And for that reason, were called satellite cells, but are also called muscle stem cells. You can see them here on the surface of an isolated single muscle fiber. So quiescent satellite cells are responsible for muscle regeneration because they are capable of self-renewal. They turn on the myogenic regulatory factor, MyoD, and when they get activated, they proliferate, and most of them differentiate and reform this skeletal muscle myofiber, but some of them self-renew and reform the satellite cell. So the outline of what I'm going to talk about is about quiescent muscle stem cells and regeneration. I'll talk a lot about a synchronized culture model that we've used for investigating quiescence. And this comes from my training as a cell biologist and an interest in working with cells in culture. And I will try and connect quiescence with adult stem cell potency and self-renewal, and talk to you a little bit about molecular regulators of quiescent cells that we have identified, which work at diverse levels. So damage releases signals that activate the quiescent stem cells. So you can see here one of these quiescent stem cells on a single isolated muscle fiber. And when damage happens, these cells grow into a little clone on the surface of the fiber. But what is it that sends activated cells back into quiescence? What are the signals that do so? And why should we care? Because most of regenerative medicine is focused on this step, which is how do you make more of stem cells so that they can repair a tissue well? But the reason why you need to know about this is because this is the self-renewal process that ensures that if muscle has a second injury at a later, future, unpredictable time, that you have a reserve which is going to contribute. So the whole process of entering dormancy is actually one which is equally interesting, and it's not the opposite of activation. One of the other reasons is that defective quiescence control may actually underlie both tumor formation and degenerative disease, which are at two opposite ends of a spectrum. If you think about it, if a stem cell cannot enter quiescence, it undergoes uncontrolled proliferation leading to tumorigenesis, whereas in a differentiated tissue, if the failure of a stem cell to exit quiescence would lead to lack of repair and degeneration over time. So quiescence itself then is an important point in the cell cycle to study. So quiescence is still not well understood. What I can say is that we know quite a bit about the transcriptional control of the specification, determination, and differentiation of muscle cells. But the details of control of quiescence are still being uncovered, and I liken it to the dark matter of the cell cycle. I'm showing you a picture of CERN there. So why do I like working with cells in culture? As far as I'm concerned, they're sublime. Taking this cell biological approach was inspired, or in a sense tells me a little bit about Al Hershey's idea of scientific happiness, which was to have one experiment that works and keep doing it all the time. Okay? So Al Hershey, for some in the audience who may not recognize the name, Hershey and Chase did the iconic experiment that actually proved that genetic material was nucleic acid. And so for me, studying quiescent myoblasts is Hershey heaven. Sorry about that. I seem to have a, yeah, maybe I'll just say next slide. So basically, the cell culture model system that we have is something that recapitulates the in vivo situation. Our cultured myoblasts can be induced to enter quiescence, where they express all the markers in the right proportion as they do in vivo. They can be activated. So you can take proliferating cells, put them into a suspension culture of a thick, viscous, jelly-like material, and you can synchronously reactivate them. You can also differentiate them. And so we use this system. It's very simple. And you can do this with mouse myogenic cell lines, with primary mouse cells, with primary human cells, and also with human and mouse MSCs. So what do these sleeping cells do? Here's a single sleeping cell marked by the green PAC7. They sleep between two sheets surrounded by laminin and dystrophin. And what we found, what they do is that they repress all of the programs which keep an activated cell proliferating, but they anticipate their fate. They remember who they are, and they create a legacy. So I'll tell you a little bit about the data that suggests that that's the case. So just to remind people that when you talk about exiting from the cell cycle, there are two forms of arrest. This quiescent stem cell is in a state of reversible arrest, which means that it can actually go back into the cell cycle after having withdrawn, and it can self-renew and regenerate. However, the myofiber, represented by this nucleus, which doesn't express the stem cell marker, is in a state of permanent arrest and undergoes differentiation and takes part in tissue-specific functions, equally important. So I'm showing you here a very simple experiment which has always astonished me. So if you take 100 or 1,000 proliferating cells and you do a simple colony forming assay and you plate them on a dish, you get something like 500 colonies, which means that 500 cells could make it on their own, right? Without the participation of signals from their neighbors. If you take a previously quiescent cell population and plate it on a dish, this is how many colonies you make. Now that's kind of counterintuitive. Why would a previously quiescent population be giving you better colonies, more colonies, than a proliferating population? So what that suggests is that somehow self-renewal is enhanced while the cell is not dividing. And there's some kind of cellular regenerative program. What might the molecular basis of that be? So going back to Hershey heaven, we've used the same strategy with different approaches. What we've done is to obtain, by different means, based on the timeline of when these techniques became available, we look for differentially expressed genes, identify them in culture, make a detailed description of their expression, functionally perturb them by either knocking them down or overexpressing, and validate key points in vivo to make sure we're not diverting too far from the. So what we found is a set of chromatin regulators transcriptional regulation, and signaling. I won't go through the details of all of these, but I'll just touch on an element of data from some of them so that I give you a flavor of what kinds of experiments we've done. So the very first gene we identified was a gene called TTP, which is tris-tetraproline, which is actually a secreted, it's an RNA binding protein that prevents the translation of cytokines, okay? Now, it was interesting to us because we started out with this, ancient looking gel type situation where we were looking for differentially expressed transcripts. And when we looked in vivo, lo and behold, this transcript from a cultured muscle cell actually marked the appropriate stem cell in vivo. So this gave us evidence that in fact the cell culture system was validatable in vivo, suggesting that the cells shared properties which were important for the in vivo situation. Subsequently, we used a number of different techniques. I'm showing you here the use of retroviral gene trapping to trap promoters which were specifically activated in quiescent cells. And then when we knocked down genes that were collected from this, we found that one of them was an architectural type chromatin factor, which actually restrains the cell cycle from re-entry. And once the cell does re-enter, it promotes differentiation. So that's another set of important. Yet another chromatin regulator, a mixed lineage leukemia factor 5, which is nuclear protein also, specifically enriched in quiescent cells, turns out to orchestrate this huge network of proteins which are all focused on the methylation status of the cyclin A promoter, a key regulator of S phase. So here you have, in quiescent cells, a set of genes which are regulating the inappropriate expression of cyclin A, and it also regulates the myogenic factors. So what it does is that it actually acts as a promoting, as a lineage promoting memory factor. So this was also obviously of interest for a dormant cell to remember that even if you've shut off lineage when you go into dormancy, you need to remember that you're a muscle cell and not a liver cell when you come back out in order to contribute the right cell to the tissue. So when microarrays became available, this is fine. When microarrays became available, when we did a detailed comparison of quiescence with other out-of-cycle states, what we found was that the genetic signature of deep quiescence suggested attributes that really promote stem cell function. Induction of survival, repair, and longevity pathways, repressing differentiation, but also determination. And this was interesting to us because it explains one of the reasons why other cell cycle exits are irreversible, whereas this reversible cell cycle exit is able to prevent all of these other pathways from being tackled. I have no idea where I'm on time, so please. Okay, so in other words, a quiescent cell has to maintain a kind of a balancing act, right? It keeps these two antagonistic programs repressed, but poised for activation because it needs to be able to activate them once it comes back. And those are the cell cycle program and the muscle program. So how is this balance regulated? So from our microarray analysis, we picked out a candidate called PRDM2. Doesn't matter what it stands for, but it's basically a histone H3K9 methyltransferase, which is essentially creating an inhibitory mark on chromatin and restraining gene function. So what we found was, very gratifyingly, the gene which we had pulled out from our culture system marks satellite cells in vivo, not just from the mouse, but also from humans. And when you knock it down, you find that it's a master repressor of myogenesis in quiescence. So if you knock it down in differentiating cells, there's hardly any differentially regulated genes. But if you knock it down in quiescent cells, you can see that there's this massive number of genes, something like 1,600 genes which were altered. And very interestingly, all the upregulated transcripts had to do with muscle function, and all the downregulated transcripts had to do with cell cycle function. Now, this was something of a paradox. So what's happening is that the cell cycle regulators seem to be hyper-repressed. They're already down because a cell is quiescent. And you're repressing cell fate regulators, but inducing myogenic regulators. So when we looked at this, we realized that this was, as I said, a paradox. And perhaps what's happening is that it actually protects the cell cycle loci from being permanently silenced. Remember, the H3K9 mark, methylation mark, is repressive, but it's not silencing like the H3K27 methylation marks, which actually sort of coalesce chromatin and bring it together in a really unreadable state. So when we did genome-wide location analysis using ChIP-ChIP and looked at all the sites where PRDM was located in quiescent cells, we found that 65% of them overlapped with the function of the protein itself. Of course, not proving that it is the protein that does the work, but at least it's a correlation. And that it associates with, by binding to EZH2 of the PRC2 complex, which is one of these complexes which is critical for silencing genes. So by preventing the, by sequestering EZH2 and preventing it from going on to, for example, a target promoter like the cyclin A promoter, which I mentioned earlier, it actually ensures that activation can be finally happen when the cell returns to the cell cycle. When you knock it down, EZH2 is free to modify chromatin and completely silences the locus. So this then was a really interesting regulator of quiescence. And what we find is that this poising of cyclin A2, which I depict here as, sort of poised on a knife edge, that this balance is really critical for the reversibility of arrest, which is necessary for the cell to return to its regenerative functions. So in other words, quiescent adult stem cells are not sleeping beauties, but they're actually high wire artists. They're doing this balancing act all the time. So in order to stay quiet, you have to be sort of doing a lot of work. It's sort of like looking at a swan which is floating on the surface of a lake, which is so perfect and serene, but underneath there's lots of work going on with the legs paddling, right? So this genome-wide poising, which was partly explained by PRDM2, we asked, does the transcriptional machinery also play a role? And work from a number of labs, Karen Edelman, Mike Levine, Jim Darnell, and Jennifer Darnell and others, showed that RNA polymerase II executes a promoter proximal pause. Once it initiates, it synthesizes a few nucleotides of transcript and then pauses. And that this transcriptional regulation actually coordinates regulation. So we asked, does this happen in maintaining quiescence? And we found that, in fact, it does. So this is just a quick picture to remind people of the actual promoter block, which is mediated by NELF and DSIF, which are two factors that come and add to this huge multi-enzyme RNA polymerase complex. And once productive elongation starts again, there's a pause release. So the questions we asked were, does this differ between select cellular states? Does it contribute to keeping the quiescent genome poised? And so what we did was we assessed the functional output of transcription. By now RNA-seq had become available. We assessed global RNA pol2 occupancy, and then we integrated these data sets to look at what kinds of genes were being regulated. And what we found was, yes, indeed, there's a massive downregulation of transcription in G0 or quiescence compared to proliferating cells, and that the stalling that we saw was associated with this repression. Not surprising. What was the fate of these G0 stall genes, these quiescent stall genes during cell cycle reentry? what we found was that they were actually very rapidly transcriptionally induced upon quiescence, and that the Pol2 marks also changed. So this increase in the active Pol2 marks is consistent with a burst in the transcriptional activity of the activating cell. And what we found was that if you knock down G0 Sol genes in proliferating cells now, it actually leads to features of quiescence, validating the fact that these genes need to be turned down in order to keep the quiescent state in a poised form. Yeah. So I won't tell you in detail about the signaling alterations in quiescent cells, but I'll tell you two things, just the titles. One is that, for example, the well-known Wnt transcription factor beta-catenin actually shifts its partner, and its target transcription factor LEF1 actually partners now with SMAD3, bringing it under TGF-beta control rather than Wnt control in quiescent cells. The other is a role for the primary cilium, which is this non-motile little antenna which sticks out of the cell. And if you knock down that antenna, remember, this is formed by the capture of the centrosome. So it's a key tumor suppressive factor. When you're taking the centrosome out of commission, you can no longer form a mitotic spindle. So if you've got it at the membrane making a cilium, the cell can no longer divide. This has been known for a very long time, but the issue was, what is it actually doing in quiescent cells? And what we found was that if you knock it down, you actually activate a whole number of signaling pathways. So it's actually restraining many signaling pathways, which feed into the TOR pathway, which is critical for protein synthesis. Okay. So then one of the things that we also asked was, which was something that we're doing right now, is, are quiescent muscle stem cells only targets for activating signals, or do they actually also signal to other cells in their environment? Are they just passive and the recipients, or can they actually signal? What we are looking at is actually exosomes, which are, it turns out, secreted quite robustly by quiescent cells, and which appear, at least from our cell culture model, these cells which are the quiescent cells secrete huge numbers of exosomes, which seem to be preferentially taken up by the differentiated cells, and vice versa. The differentiated fibers release exosomes which are taken up by the quiescent stem cells. So it's almost like there's a crosstalk going on between two components of a tissue which were formed from the same developmental origin, but they're actually modulating each other's function. And one of the things that my last PhD student is doing is trying to figure out how this happens. Okay. So in other words, I've been in Hershey heaven for 27 years. Quiescence biology revealed by all these studies shows that they have profound overlaps with quiescent muscle stem cells in vivo. It's not a sleeping state, but it's poised for activation. So these are just the different aspects of quiescence. I think I've belabored the point too much. So why is it that this might be important? Why is understanding what happens to quiescent cells in vivo important? Because stem cell transplantation for things like muscular dystrophies is completely impractical. You have a huge dispersed target. How are you going to inject? First of all, you're not even going to be able to get enough cells to be able to service something which is degenerating body-wide. So the idea is that we would think of ways to prevent degeneration, or ways to actually understand how homeostatic repair happens, so that we can target these endogenous muscle stem cells to repair diseased muscle. For that, we need to understand a lot more about molecular control of quiescence, so that we can sustain these cells in a self-regenerating, potent form. So the dreaming cell. It has issues, it plays an important role in identity and legacy in cells and in tissues, but also in institutes. I think there are metaphors for So this is my favorite cartoon. I love Kung Fu Panda and Master Shifu, who represent stillness and balance. And basically what we've found from our work is that these metaphors from life actually inform the biology of these cells. Anticipatory programs govern the quiescent stem cell state, and they're critical for tissue repair. So metaphors from biology also inform science organizations. Institutions need to develop, they need to grow, and they need to regenerate because we cannot maintain the same momentum over time. So many years ago, Dr. Bhan made me an offer I could not refuse, and got me involved in working with a group of people in Bangalore to help set up InSTEM, which has now grown to a very large unit integrated with NCBS and CCAMP and TIGS. So the Institute for Stem Cell Science and Regenerative Medicine, now headed by Manisha Inamdar, is a wonderful place because it had the mission was collaborative work. And this cooperative environment that it has fostered has enabled people to work together in teams in such a way that they can tackle problems which are really beyond the scope of a single lab. And it's integrated many other people. So here's a slide from Manisha Inamdar, who's the director now, telling us about all the different, can we go back? The different aspects of work going on at InSTEM. And anyone who's interested can look at the website. So just to leave you with a few thoughts, working with your hands is not separated from the mind. Deep descriptions of a biological process are necessary before we can have any kind of mechanistic understanding. I show you here the beautiful pictures of Ramon Cajal, which he drew by hand, by simple, pure observation and detailed description. I'm sure all of you who are more senior would identify with this issue. Can we go back? That a life in science is this constant seesaw between discontent with what we already know and a satisfaction at some small inroads into the unknown. And so I think often of Ramon Cajal with his beautiful descriptions which paved the way for molecular neuroscience and a deep understanding of the brain. Now, what happens, I think, at many times in our career, especially when we're younger, but even when we grow older, we somehow worry that we are not being recognized enough. We worry that, our Am I audible? I guess not. Yeah. This one. You'll have to turn it on. Okay, cliffhanger. Okay, so as I said, okay, sounds like we're in business. Are we? No. Yes. Okay. So, as I said, they're determined not to let me philosophize. I'm trying to, yeah. It's still not on.
There's a technical glitch that has happened.
Okay, now I'll really do the dance that I said I would do.
While the microphone comes back.
Yeah.
Can you fix it? No. Power, yeah. Okay. We need to go back.
Hello.
Okay. Needed him to come. All right. Am I audible? Not really. He needs to stand beside me, maybe. Hello? Hello? Not really. Seriously, I think all this talk of sleeping stem cells put the mic to sleep. Yes?
No, we are still awake.
You're still awake? Okay. Am I audible? Okay. So there's not much left. I just wanted to say that when we have moments of anxiety about the fact that we're not being recognized enough and that people don't really understand what we're doing, etc., it's very fruitful to me to think about Gregor Mendel, who did these path-breaking experiments in a small town and presented the results to an audience of 40 people on a cold winter day. And this set of 40 people listened and nodded their heads and did not understand what he was saying. But the paper was published in the Proceedings of that society, the Scientific Society of Brunn. He never knew that his results had the impact that they had. He never got any credit in his lifetime. He made 40 reprints and he sent them to 40 people on the continent who were great scientists. He sent one to Darwin. Darwin never read his copy because that copy was found uncut in his study. So they knew that he had it, but he never read it. Just imagine what might have happened in that period had Darwin read Mendel. Anyway, all was not lost. He was rediscovered, and it transformed our understanding of inheritance and paved the way for the evolutionary synthesis as well as the molecular evolution. So anytime you're feeling depressed about not being recognized, just remember Mendel. Okay, last slide. When I say we in the lab, it means they, all of the wonderful people who've worked with me over the years. And I want to end just by thanking my collaborators and funding, and in particular, CCMB and INSTEM for being warm and hospitable environments to do work in a little backwater of quiescent stem cells. Thank you very much.
So can you stay here? We will be felicitating you. Okay. Thank you very much, Dr. Dhawan, by presenting the beautiful world of quiescent cells, and thank you for closing your presentation with a good message. You know, sometimes we just get disoriented that kuch kaam nahi ho raha hai, kuch chal nahi raha hai. Reviewers are rejecting our papers, grants are getting rejected, but yes, we'll continue doing our work and try to excel in our work. Thank you so much. And with this, I would like to invite Director and Professor Ashutosh Sharma to come up on the stage to felicitate Professor Dhawan. Yeah. Good morning.
How are you?
I'm good.
All
Doing
good?
good. Yeah. Nice to see you.
So you're going to fix this up?
It
By
would
the
be
neighbor?
definitely. Yeah. For the photogenic photo. So Dr. Dhawan is here with us the whole day and till the evening as well as at night. So I know there would be a lot of questions. So you guys can catch hold of Dr. Dhawan, and Dr. Ashutosh Sharma is also here for a while. So we will break for tea, and in half an hour we'll come back again. And I request the colleagues who would be felicitated, please take your marked seats in front. Thank you. Nice meeting you. I've heard you on a couple of occasions. I'm Neeti Ayub.
And actually, you know, in Niti Ayog, Vice Chairman used to be one Rajiv Kumar. So you are actually a hybrid, Niti and Kumar.
Actually, his
That fits
wife's
in.
name is also Niti.
Yeah, that's true. But double E, you are one I.
There's some difference. Okay, very good.
I could
join
not
us.
attend the INEAS meeting
No,
because
no,
of,
no, no,
I
no,
knew
no.
that you were coming from there.
Yeah, yeah.